This podcast is not meant to be professional advice of any kind. It is meant to be informative and entertaining. If you make any changes to your life, see the appropriate professional before you do so. Hello and welcome to Super Age. My name is David Stewart. I'm the founder of AGEIST. At Super Age, we help you live better and become the best version of yourself. And who doesn't want a Super Age? Welcome to episode 99 of the Super Age podcast. This will be dropping on August the 31st, 2022. So today, as we're working on this podcast, I'm looking out the windows next to where I live here, and I have this sort of large shrub, small tree thing that's covered with berries. And it's right next to the windows. So what happens at this time of year, every year, towards the ends of August, the berries all they ripen. So they turn this sort of like dark purple color. And then they become this feeding magnet for the birds. And it's this, I wrote a little bit about this in the newsletter today. I just love this time of year because you get to see, it's like Wild Kingdom. Remember that Mutual of Omaha, Wild Kingdom? It's like that right outside my window. And I have all these different species of birds applying various strategies, I guess, to get the berries. And initially, it's really easy for them because the berries are sort of, they go for the ones on the thick branches first. But then as those get sort of weeded out, they have to get themselves out on these spindly little ends and grab the berry. And sometimes that works out and sometimes doesn't work out so well. But I'm watching this right now, this sort of medium-sized green bird like chow down on berries because, winter's coming and, build up the fuel store. So staying on the animal theme, something else that I've become very interested in recently is something called animal movement, which is not the movement of actual animals across the landscape. What it means is us humans mimicking the way animals move on the ground. And so it's a lot of sort of ground-based movement patterns. There's bear walking and gorilla walking and backwards crab walking and some moves that sort of feel like they're breakdancing in some way. And, I'm pretty fit and I'm really, really strong. But this is like a whole other thing. It's sort of a coordination thing, but it's more the sensation. I got to tell you. So the sensation is sort of like adult romper room. Like you're on the ground all the time. Either your butt's on the ground or your feet and your hands are on the ground or you're doing these movements. And it's just so playful. Like I have to say, when I started doing these, the guy who was teaching me these, it was like, wow, these are hard. But then I sort of got the hang of it, and now it's just I can't wait to do it again because it's this really playful movement release, and it's sort of like dancing on the ground, but it's I think because of these animal movements, it's like wired into the back of your brain in some funny primitive way that says like, oh yeah, this is cool. I don't know. So check that out. There's a lot of information online about animal movement. I've become a big fan of this. I hope you enjoyed it. If you like it, let me know. And on that note, you can hit me up, david@superage.com. Great to hear from you, whatever's on your mind. And also, if you're enjoying this podcast, you have the opportunity to leave us up to a 5-star review wherever you're listening to this and to leave us a comment. We love comments. And maybe share it with a friend because that's the only way we grow is all of you out there tell your friends about us and, you say, this is cool, you should check this out. This week on the show, it's word of warning, there's going to be a lot of science. We often have science on this show. This time, we're getting deep into it, so prepare yourself. But it's awesome what we're going to talk about here. We have Momo Vuyisich. I'm sure I just destroyed his name. Everybody calls him Momo. He's got a PhD in biochemistry, and he is the co-founder and chief science officer and head of clinical research at Viome Life Sciences. Viome is one of the companies we work with, so full disclosure there. But I wanted to have Momo on because I've taken a couple of the Viome tests, and it's really extraordinary. I sort of understood how they work, but not really. So I wanted to have Momo on to explain to everyone what's going on here and how, in his mind, this is health 2.0. This is the next way that we're going to be personalizing our health. And his strategy involves some DNA work, but it's mostly RNA work. And I'm going to let him explain how that works. And I think that you're going to find this fascinating. I think that, come in the next few years, this is how pretty much all diagnostics are going to be done. And you'll be able to be diagnosed. You'll be able to be personalized, your nutrition, your exercise, all of that based on this RNA technology. So let's give Momo a call. Hey, Momo, how are you?
Doing amazing. How are you?
I'm doing great too. I'm so excited to have you on here. As we get started, tell me a little bit about you're with Viome Life Sciences, and what do you do there?
I'm a co-founder and chief science officer, and I also lead our clinical research programs. And then, there's additional thousands of different tasks that an officer would be doing for a company like this.
So, as we get started, I want to tell everybody the reason I'm having you on is because I took the Viome gut test back a few months ago, and I'd been having really terrible gut issues for a couple of years. I'd ended up in the ER a couple of times, and I'd done all the parasite tests and the food allergy, food sensitivity, all these things. And nobody, they were just like, well, we don't know what's going on. And I took the Viome test. And then what happened was I got it back, and there were about 10 or 12 foods that I should not eat because my gut contained plant viruses that were causing my gut dysbiosis. And I stopped eating those foods. And 3 days later, my gut was not entirely normal, but 50% better, just like that. Boom. So since you are the origin of that, I just want to say thank you. my gut's happier because of you. But so what I want to learn is, what is this technology? How are you able to find these plant viruses that seemingly nobody else looks for? How does this all work?
How does this all work?
Yeah, please.
Yeah. So it's very complicated, but I'll try to stay at a high level and make it simpler. So I would like first the audience to understand the difference between DNA and RNA. So DNA is something that people have been talking about and focused on since the Human Genome Project 20 years ago. And by sequencing DNA of humans, the scientific community promised that, 20 years ago, that we will address all chronic diseases and cure all of them and all that stuff. But that hasn't happened. And the reason is because now we know that genetics only provides a risk. It doesn't actually know what's going to happen. It has the potential, but it doesn't actually execute on anything. DNA is just a static molecule that sits there and doesn't do anything. What actually executes the functions of DNA are RNA and proteins. Now, proteins are very challenging to measure, and they're not. Measuring the proteins is not something that you can scale as a consumer business. That's a very advanced laboratory technique that doesn't scale. So we decided to develop an RNA detection technology. So we measure RNA. All of our tests currently analyze RNA. And RNA comes from every single living organism on the planet, including viruses and humans and bacteria and everything else. So that's one really important thing, that every living organism has RNA. The second important thing that ties to what you said is that there are, in fact, RNA viruses that do not have any DNA whatsoever. And so, for example, plant viruses, and then, for example, SARS-CoV-2 that causes COVID, or many other human viruses like influenza, they do not have any DNA in them. So if you analyze DNA, you cannot possibly see them because they do not have any DNA. And so the advantages of our technology are that we actually sequence all the bits and pieces of every microorganism, so we can identify all microorganisms. And I know you were curious about how many we see, and I have the statistics actually here. The average person's stool has 1,300 different microorganisms, And that includes bacteria, fungi, archaea, and viruses. And there's all kinds of viruses. There's human viruses, there's plant viruses, and there's bacterial viruses called bacteriophages. So 1,300 unique different kinds of microorganisms. And I think everyone by now knows that there's 40 trillion of them total. There's 1,300 different kinds. And then the total number of microbial genomes that we've identified so far in all of our 400,000 samples is 16,500. So those are the stats. And so we can see all of those microbes and we can identify them, but that's really not the main advantage of our technology, because identifying that you have a microorganism, all that it tells you is that, oh, you have these microorganisms, you can be healthy with them or you can be sick with them. Okay. So for example, an example that I like to give is an IBD patient. An IBD patient has a static microbiome in terms of composition in their gut. And so they have the same microbiome when they're in remission and when they're in a flare. They also have the same human genome. Their genome does not change during the periods of remission and flare. So as a person with IBD goes through remission of several months, meaning they have no symptoms and they're healthy, and then they go into a flare, and now for the next month, 2 or 3, they're super sick and they cannot function, their microbiome does not change the composition. The same microbes that were there a month ago are still there today. So it's not really who is there that determines whether you're going to be healthy or not. It's what they're doing. And so an average, let's say, microbe in the gut codes for 3,000 genes. And every one of those genes has a different function. And so the microorganism will only utilize some of those genes in different environments. And so I'll give you one extreme example. We have a bacterium called Clostridioides difficile. So everyone knows it as C. diff. This is a bacterium that's known as a pathobiont. And a pathobiont means it's a bacterium that normally lives inside human beings. So 42% of our customers have C. diff in their stool. And it's a very healthy bacterium in that it produces butyrate, and it's very healthy, and we've evolved to live with it and to depend on it. But if we consume antibiotics and we kill off the other bacteria, then this bacterium, the same bacterium, can turn into a pathogen and kill the person. And so it's the same genome, meaning you can sequence the DNA of this bacterium when it's friendly and it's helpful, and you can sequence the genome when it's actually a pathogen, and it's the same genome because it's the same bacterium. It carries the same genome, but that genome has the potential to be helpful or to kill you. And it's the genes that are expressed that make a difference in this behavior, and that's what we measure. So we measure the gene expression and not the genome potential. Does that make sense, or is that pretty technical?
Sort of makes sense. So the RNA is the expression of the gene?
Yes, exactly. It's which genes are active and to what degree. And so, I mean, think of trying to sequence someone's genome and say this person is a criminal, or think of trying to interpret genomic information as behavior. You cannot do that because the behavior changes depending on the environment. A person can be absolutely lovely in one environment and absolutely hateful in another environment, and a criminal in a third environment, depending on what environment you put them in. They have the potential, their DNA has the potential to express all these behaviors, but only a certain type of behavior is expressed under certain conditions. And that depends on the environment. So you have to measure the environment and you have to measure what the microorganism is doing.
So these DNA tests are probabilities.
They're possibilities. They're possibilities. What is all possible? The DNA test, for example, can tell you, you have this C. diff, and you can be either very well nourished with butyrate, or it can kill you. That's what the DNA test says. And that's all it can say. It cannot go beyond that because it doesn't measure what that bacterium is actually doing. Is it killing you, or is it actually nourishing you at this point in time? Because it can do both, or either, or none.
So we have this incredibly complex environment. And so you've got 16,500 of these viruses, fungi, bacteria, all this stuff, and they're interacting. Help me to understand how you register what's how they're interacting well or they're misbehaving and you get dysbiosis. How are you telling that?
Yeah, so we're telling that by looking at their gene expression and understanding. So let's back up one step. Okay. So first, I would like the audience to understand these functions. So let me give you the most simplistic possible thing, and that is, how do you make alcohol in beer? You provide a single microorganism, typically brewer's yeast, right? Which is Saccharomyces cerevisiae, and you provide it with substrate or food, which is barley, which is a polysaccharide fiber, and it uses that polysaccharide to digest it, and as it digests it to create energy for itself so that it can grow, the byproduct of that metabolism is alcohol. The yeast does not consume alcohol. It's literally its waste. It's what I call micropoop, right? So if you just think of those three pieces, you have barley, it's being processed by a live microorganism into ethanol. Okay. That's in chemistry, that's like A goes to B, right? Via an enzyme. These enzymes that are converting barley into alcohol, they're enzymes encoded by the genes of the yeast. So they're little nano machines that are doing chemical processing. They're like chemical factories, and they're making that. So think that now each bacterium in your gut has 3,000 of these functions, and you have 1,300 microorganisms, right? So there's about 3 million functions encoded by a typical microbiome, 3 to 4 million functions, right? But of those functions, at any point in time, only tens of thousands are actually working, and we can measure which ones are working. So that's one thing, right? The second thing is, in our clinical research, we can identify which functions are good for you and which functions are bad for you. Okay. So let's just make the simplest possible case, which is very well known. It's not something we discovered. So we have butyrate as a metabolite or a product of bacteria in the colon that is known to be super healthy, right? And there's lipopolysaccharide, which it's a diverse number of molecules, and some of them are actually very good for you, but let's just bundle them all into, for now, for just argument, that LPS is bad for you, let's say. So everyone has bacteria in their, or microbes in their stool that can produce butyrate and that can produce LPS. But there are different kinds of bacteria, and they take different inputs. Some of them would feed on barley. Others would need artichokes or apples or leafy greens or broccoli, or different bacteria consume different nutritional ingredients to produce the same beneficial or harmful chemicals in your colon. And so now, when we measure who is there in your stool and what they're doing and what they're capable of doing, our computers can actually prescribe exactly which molecular ingredients your microbiome needs, and emphasis your microbiome needs, in order to produce the beneficial chemicals, just like that yeast was producing alcohol from barley. On the other hand, once we know which of your microbes can use which molecular ingredients in foods to produce bad chemicals, we now put those foods into avoid foods. We basically deprive your microbiome of the ingredients that it needs in order to produce bad chemicals. And that's how we control the functions or the activities of the microbiome for every person. And if you go to another person, their gut microbiome is different. And so they're going to need to consume and avoid different foods because their microbiome is different. So going back to my simple example of yeast, right? If you do not give barley to that yeast, you're not going to get your beer, right? And so if you tell the person who holds that yeast, hey, stop eating barley, there's not going to be any alcohol, because if we know alcohol is bad for you, that's how we control the functions of the microbiome, by simply either depriving them of the molecular ingredients in foods, because our microbiome feeds off our food. So we are feeding it. And so if we control that, we can control what the microbiome is doing, and we can shift the functions of the microbiome from whatever the balance is at the moment, which could be really bad or pretty good or whatever it is, towards better and better and better. Does that make sense?
Super complicated. And I'm glad you figured it out, because I couldn't.
Yeah, it's super complicated. I mean, I have a PhD in chemistry. So to me, this was basically what we were taught and what we understood. It's just much more complex than like simple reactions because there are so many different reactions at the same time. But there are two things that have enabled this, two technologies that have enabled this. One is the next generation sequencing. So next generation sequencing has accelerated sequencing by 1 billion-fold over the last 20 years. That's very significant. And the next one is supercomputing. And so right now, as you and I are speaking, we are actually running 3 very large supercomputers in the cloud. They're running 24/7. And so these kinds of technologies where we can generate vast amounts of data and process them did not exist just a few years ago. And one example that I like to give is that if you let's say you go to a doctor and they prescribe to you like a lipid panel, and you go to get phlebotomy done, and they draw many milliliters of blood, and you get 4 data points out of that test. You get HDL, LDL, cholesterol, and triglycerides, right? 4 data points from milliliters of blood. Our blood test uses 50 microliters of blood, which is literally 1 drop. And we produce about 20 to 30,000 data points from that with our test, right? And so the technological advances are extraordinary relative to old school technology, which was just a few years ago. And while we're on that test, I know some audience members are going to say, whoa, 1 drop of blood. Wait a minute, that sounds too good to be true. So when we developed our test, because of Theranos precedents and all that, we clinically validated that test. We clinically licensed it with CMS. So they come and inspect our lab, inspect our quality control, inspect our results. So this is an actual test that exists. And we published it in a peer-reviewed article so that this test is super, super legit. It's not something that we are selling or pitching as a hypothetical thing. It is being performed today in our lab every single day. And it's clinically licensed, and the US government inspects it on a regular basis to ensure its performance.
Yes, I mean, you've made a couple of clear points there. You are not Theranos,
right?
You're not doing this test in some little box that's going to sit on somebody's shelf. You have a massive amount of computing power and sequencing that is causing this to happen.
Yes, exactly.
Yeah, amazing. So you've got a new test out, which I haven't done yet, which I'm going to do. The oral biome.
We have 2 new tests in the last month.
Oh, please tell me. What's the other one?
Yeah, so both are saliva tests.
Uh-huh.
I don't know if the audience knows, but we've had for a long time now the stool test only, which is called Gut Intelligence, the stool and blood tests, and that kit is called Health Intelligence. And now we've added a saliva test for 2 purposes. One is we've added it to the stool and blood combination, and it's now called the Full Body Intelligence. And that means that a consumer today has access to our metatranscriptomic technology that the vast majority of research scientists do not have access to. It's so advanced that the vast majority of the publications today do not utilize this technology because it's too expensive, too complicated. Very few places can actually attempt to do it. So that's a consumer product whose output is wellness, which means that customers get results and recommendations, and the recommendations are 100% nutritional. So it's food and supplements. So that's on the nutritional side, on the wellness side. But we're also very proud that we have now created our first diagnostic test. So this test is detecting the presence of oral or throat cancer. So it has 95% specificity and 90% sensitivity, and that includes stage 1. And this is really something that we're super proud of because it's not well known that even though oral and throat cancers are not very common, they are detected in late stages 3 and 4, which have a very high mortality rate. But even if the person survives, there are essentially lifelong consequences of the treatment because the surgery takes out a lot of the parts. And so they're unable to chew, for example, they're unable to speak, and so on, many of them. And so it's devastating to get that. And so there hasn't been a technology that can detect cancer in stages 1 and 2 that are very treatable. And our technology does that. So the specificity and sensitivity numbers I gave you apply to all 4 stages of cancer. And that's super important. And that's one of the main reasons that the FDA reviewed this and gave us a breakthrough device status. And so now we're moving through the so-called pivotal trial, which is a device trial that the FDA will review and either give us clearance or not by next summer or so. But in the meantime, even before the FDA approval, we've launched this as a clinical test. So just last week, we launched this and people can order it on our website. Basically, when a customer places an order, we have a telehealth provider that reviews the order and makes sure that the customer is eligible for the test. They approve it. The customer is shipped a saliva collection kit. They collect saliva at home and ship it back to our lab where we analyze it. If the result is negative, that there is no oral or throat cancer, the customer receives that result directly as a negative. If the result is positive, that result is delivered to our telehealth provider. Then the doctors from there reach out to the customer and start taking care of them in terms of follow-up, in terms of biopsies and additional tests to provide proper health care.
You said if a person is eligible, what are the eligibilities?
Yeah, so currently there are two groups of eligibility. One is a person has to be 50 years or older. That's it, just 50 years and older. Anything else. The second is anyone 18 or older who has a history of tobacco use, and that can be cigarettes or chewing tobacco or vaping. Any kind of tobacco use increases your risk significantly of oral and throat cancer. Those are also eligible currently. Yeah.
I want to go to the oral biome test.
Okay.
Why do I need to test my oral biome?
Well, oral microbiome is turning out to be just as important as the gut microbiome. It's just that for some reason, the gut microbiome was made famous initially, but oral microbiome is super important. So I'll just give some flavor of that. So for example, the best test for autism spectrum disorder is in saliva. It's measuring RNA for human and microbial RNAs. The same with Parkinson's. The bacteria that have been associated with Alzheimer's disease, two of them, Treponema denticola and Porphyromonas gingivalis, they're both normal residents of the oral microbiome. They occupy the oral, the oral cavity in many people, but they can turn pathogenic. So this is another example of these pathobionts, where they normally live in the oral cavity, but then they can, via inflamed gums, they can inflame gums, they can translocate into the blood and eventually translocate into the brain and cause Alzheimer's disease. But in the recent publications in the last year or so, oral microbiome has been connected with things like arthritis, with things like insulin resistance, which then leads to obesity and diabetes. And so we're just starting to discover, the importance of the oral microbiome. It's also the reservoir for a bacterium called Fusobacterium nucleatum, where that bacterium, when it travels to the colon, it actually causes or at least enhances colorectal cancer. And so, those are just some of the main points.
Wow. So now you've got two very complex microbiomes that interact.
Yes.
That sounds like a bit of a data challenge.
Yeah, it's a huge data challenge. And so they interact in a couple of ways. In one way they interact is that people who take antacids, especially people who take PPIs every day, and they weaken their acid. The stomach acid is there not only to digest food, but to prevent microorganisms from flooding the intestines. And people who are on PPIs or antacids, they tend to have more of the oral microbial species in their intestines, which is not good at all. For example, Fusobacterium nucleatum is very well known. So that's one connection, direct basically translocation of oral microbiome into the intestines. The second connection is more of a systemic. So I would like people to think about Viome Life Sciences as a systems biology company. And systems biology just means that no matter what condition you're looking at, you should look at the whole body and not just the isolated organ. So for example, we've learned just recently that people who have Alzheimer's, the brain lesions that are observed in the latest stage of Alzheimer's are just one of the organs affected. They likely have a gum disease. They likely have chronic inflammation. They likely have an impaired blood-brain barrier, and they likely have leaky gut. And if you look at almost any other disease, it's systemic. There is an involvement of the immune system, of genetics, of oral and gut microbiome, and food, right? Nutrition and heavy metals, molds, mycotoxins, all these things. And so we should look at everything as a system from the systems biology perspective. And that's why we've introduced this sort of a system. We've been doing this for clinical research, stool, blood, and saliva. We've been doing that since day one. But now we've made it available to every consumer because there's so many interactions.
So I haven't taken the oral microbiome test. I will. But assuming I take that test and there's something out of balance, I understand with my gut, eating certain things would be superfoods, avoiding other things. But my mouth, what happens there?
Yeah, that's amazing. Well, we already know that while you're chewing food, the process of digestion starts in the mouth. So there are enzymes that are released in the mouth, and there are both microbial and human enzymes that are released in the mouth to start the process of chewing food. So that's one thing. The second thing is that I'll give you one specific example. There are nutrients that are in fact digested and produced in the gut, but they travel via bloodstream and they're actually secreted in the oral cavity. So nitrates and nitrites are very important ones. So for example, we can modulate the oral microbiome by eating foods that contain nitrates. For example, beets are the most well-known source of nitrates. But if you eat beets, the nitrate in the gut microbiome is actually converted and then I think into nitrites, and they travel to the oral cavity and are affecting the oral microbiome. So that's currently where we are, but we are already working on lozenges. And so we are identifying right now all of the best ways basically that we can modulate the oral microbiome to go from not healthy to healthy, just like what we've been doing with the gut microbiome. And so we are currently formulating lozenges that we will provide to our customers, and they will have prebiotics and probiotics. And so you will be sucking on those for a long time and basically providing not only new probiotics to seed, to colonize your oral cavity, but also prebiotics to feed good members of your oral microbiome. So we're moving in that direction to actively modulate oral microbiome.
And the whole nitrite, I always get that confused, the nitrites and the nitrates that come up from your gut into your saliva. And that, if I have to get this right, interacts with bacteria, which produces nitric oxide in your blood. Did I get that right?
Yeah. Nitric oxide is also produced by our enzymes, and it's produced by bacteria. But I think the reduction of nitrate to nitrite is actually occurring in the mouth by oral microbiota. And that in some cases is beneficial, in other cases it's detrimental. And that's another reason why you have to look at the, nitrous oxide is good for some people, not good for some, or good under certain circumstances, not good for others. And that's really this whole precision medicine movement that has happened over the last 15 years is really, there's no blanket statement. For example, we talk about, E. coli is bad, or there's this microorganism called Akkermansia muciniphila, and we talk about it as being good. But both of them have the potential to be good and bad. And both of them can have, E. coli can have very healthy outcomes, and Akkermansia can potentially have very bad outcomes. It's been associated with Parkinson's disease and some other conditions, multiple sclerosis specifically. And so we just have to be careful to not judge these microorganisms and not label them as good or bad, but to look at all the potential functions that they could have and then eliminate their bad functions and stimulate their good functions. That's really what this is about.
And help me to understand a timeline. Okay. So I've been following the recommendations religiously. You go a couple times to the ER, that gets your attention.
Yes.
You follow directions. So I've been doing this for a few months now. How long would it take? And what I'm just sort of guessing here is we eliminate the first 10 or 12 bad ones, and then by starving them, I guess, essentially.
Yeah.
And then how long before those are cleared up, and then I should probably test again, because my guess is there's probably other ones that are in there.
Yeah. So we currently recommend retesting every 6 months as sort of a generic rule for everyone. But the actual truth is that it depends. And so, for people who are making minor changes to their gut microbiome functions, and people who are not eating foods with chemicals in them, like preservatives and antibiotics, things like that. Those tend to have very stable microbiomes on a longer scale, and those people probably don't have to test more than once a year. Then there are those people who are consuming, unfortunately, lots of antibiotics, whether via food or whether via actual, antibiotic consumption, and lots of preservatives that are actually destroying their microbiome, and they may already have a weakened microbiome. Those people are going to be experiencing much more rapid changes, and for them, every 6 months is not going to work. But again, for a typical person, assuming that people are eating relatively well and they're not taking antibiotics, then every 6 months we think is the most sort of the best value for everyone overall.
So this amazing technology that you have, where are you looking to go with this? You've already you discovered this.
So this is actually the most exciting part about Viome Life Sciences that I hope that most people will be super excited about and will be a major takeaway. So when I envisioned this technology and this platform, back in 2010 as a government scientist, I wanted to create a technology that is this advanced and molecular and super hard to do, but that would be accessible to everyone in the world, just like the App Store. And so we've basically modeled our entire company similar to the App Store concept. And so, in fact, our technology platform is called ViOS. It's not iOS, it's ViOS, the Viome operating system, right? And this technology powers all the applications. So Viome.com products that we discussed today, like the stool test, the saliva test, the cancer test, all of those are applications of ViOS. And we're currently working with many partners to generate many additional applications. So the vision is that 10 years from now, Viome Life Sciences will be a company like Apple, where we hold this ViOS under, we are developing it, we're improving it, but we're giving access to the whole world to it. And the whole world can actually be developing applications. So just like today, the App Store has 2.2 million applications, where Apple only developed a few hundred of those. I'm hoping that in 10 years, there will be 1,000 health applications developed using ViOS, but only a few of those are developed by Viome Life Sciences strictly, that the vast majority of those are developed either by partners alone or in partnership with them. So, we have an application process where anyone can apply for access to our platform. And in fact, the oral and throat cancer test was in fact developed using this process. Queensland University of Technology scientists said, wow, this is the best technology for microbiome analysis. We think that microbiome is going to be a very good signature for oral and throat cancer. They applied for access to our platform. We enabled them to succeed. We co-developed it. And now we are actually marketing the product. We're performing the tests, and they get royalties from every sale we make. We're working with some other companies where we're hoping that we will help them develop products, but they will then market those products and make the sale, and we will collect the royalties. And so that we're trying to create an ecosystem where anyone, even individuals, can actually create either products or companies that can leverage our ViOS technology to spin out human health applications of the next generation, which is focusing on preventative and curative strategies, and not these maintenance strategies where you're given medication for the rest of your life and you still are deteriorating or you're still not getting better, right? That is the mission of Viome, is to prevent and cure diseases, not to manage them and sort of keep them there. How cool is that?
That's amazing. I have to say, one of the things that amazed me when I got my results back was, like, you've used the word precision a couple times, the precision of the recommendations for the, whatever, like the NAD, nicotine riboside, like it was like super precise. How much vitamin C? Super precise.
Yeah.
It sort of shocked me.
So everything that we do at Viome is computational. So all of the recommendations are computed 100% by mathematical equations. So this is one of the things that I pride myself a lot in, that we've created what I call Nutrition 2.0. And Nutrition 2.0 is a complete quantum leap over the current nutrition. So today's nutrition is either focused on, hey, you're deficient in iron, take more iron, or hey, ketogenic diet is the best for everyone, or Mediterranean diet is the best for everyone, or whatever the diet of the day is, right? And so Nutrition 1.0 has basically very little science to it. Nutrition 2.0, which is what we've created, is 100% objective. So there is no human being involved in this whatsoever. So when the data, so we let's say we analyze someone's stool, blood, and saliva samples, the machines generate objective molecular data. These are the clinically validated tests that the US government inspects and makes sure that they're actually accurate and precise and have a linear dynamic range and all that. They create the data and send to the cloud. We overlay those chemical data with mathematical equations that compute everyone's diet and supplements. It's a computational exercise. And now these equations change over time as we learn. And so, for example, there is no literature to show that RNA viruses that come from plants have an impact on human health. There's very little literature on that, right? But our machines picked it up and said, hey, people who avoid these viruses, who have these viruses, they tend to have these problems that are similar to IBS. And people who avoid those, they don't tend to have that. And so the machines picked that up. It's not us. But this is from large amount of data. I'm the head of clinical research at Viome, and we've recruited so far 16,000 clinical research participants. We have a hyper active clinical research program because we continuously need to feed the machines more and more data so that they can come up with continuously more and more accurate algorithms for every kind of a symptom you can think of.
Okay. So let's feed the machines more data. So let's think about, rather than, I'm just going to make this up. The data that's going in right now has to do with, the things we've talked about, my gut, blood, things like that. So what happens if you put in other things, or can you put other things like, okay, this is my weight, this is my shape, this is my grip strength, this is my VO max, putting in those things. Is that something that you could see happening?
Yeah, so we actually are working currently with Apple to be able to import the Apple Health Kit data into our database so that the machines can actually utilize that data and see whether they factor into any kind of symptoms. So we are a data science company, and we'd like to get all data because we don't want to pretend to be smart enough to hypothesize whether like VO max is important for type 2 diabetes or not. I don't even want to pretend to be smart enough. I don't care whether it is or not. But what I do want to know is it or not in a data-driven fashion? So let's get the data, let's do some machine learning and find out, is it important? And if it is, for which conditions? And how do we factor that in, right? And so, we want to gather as much data as possible and see what matters for every condition. And that's how we discover things all the time. That's why we run supercomputers, because the amount of data we're generating is super vast.
Yeah, super vast. I think that, some of the clinicians that I've been speaking to, their issue is the fire hose of data. How do we manage all this different stuff coming in and make sense of it?
Yeah. Well, I mean, you need supercomputers, you need data scientists, you need, it's crazy complicated. I mean, this is the most complicated thing in terms of computational science. You also need to generate the relevant data. When I hear from cardiologists, for example, that 50% of the cardiac patients who come in with a cardiac event to the emergency room have a normal lipid panel. You have to ask yourself, why are we still using the lipid panel as a routine test if it doesn't really do anything, right? Or it doesn't do much. And so we have to go after a new data stream that is more relevant to diseases. And that's basically why we created the technology and why we created the company, is because we are very confident that the data we are generating are relevant to chronic diseases and cancers and aging. And the data so far have been limited by what we can get and not what's relevant. It's very easy to measure the lipid panel, but just because it's very easy to measure doesn't mean it's important.
I had no idea. So my LDL really is only a 50% predictor of a cardiac event.
Read about it, please. It's quite amazing that it's not a very good predictor at all. Yes. Yeah, and in addition, even if you have a high cholesterol, what are you going to do about that? You're going to eat less fat and less animal products? That's such a gross oversimplification of the problem, right? And many people do that, but our liver makes cholesterol. And so there's other things at play. It's not just, it's not just the diet, and it's not just reduce cholesterol. There's lots of other things. For example, how much degradation of the bile acids is occurring in the gut and so on. So it's very complex, and we really have to approach every problem from the systems biology view.
Wow. My head hurts just thinking about this.
It's fun. It's fun.
Amazing. So do you foresee a point working with partners and with this platform? I hadn't understood the platform strategy.
I know.
So would you foresee a time where someone comes to you and they say, well, this is very interesting technology. We think this is predictive of, some other kind of cancer, some other kinds of diseases. Is that what you're getting? People are coming to you with?
Yeah, that's already happening. We are already partnered with partners to develop novel tests. For example, for colorectal cancer, we've already partnered with GlaxoSmithKline. So yeah, we have already established several partnerships, and we're working on those right now, but we're also working on establishing new partnerships on about 20 different diseases. And so we are pushing as hard as we can to utilize ViOS for additional applications. Right now, we're working in IBD space the most. We're focused the most in IBD space, but we have programs, like I said, in 20 different diseases. So we're going to keep moving, and we welcome everyone to come and utilize this platform, and they can utilize it in several ways. They don't have to have money. They can, for example, Queensland University of Technology didn't give us any money, but they gave us patients. So we now have several hospital partners who don't give us any money, but they give us access to their patients and medical records. And that's super important for discovery. But then we also have partners who give us money. So the relationship, it can vary. The objective is that whatever they can do to complement us in order to utilize ViOS to spin out additional health applications. That's always the key objective of any partnership.
Okay, personal question. So Naveen told me he intends to live to 200.
Okay.
You're the data scientist. How long do you think you're going to live?
I don't know that.
Not predictive.
Not possible to predict right now. So yeah,
okay.
Naveen is going to likely live a very long life, but I don't know how long. So yeah. But we are working. We do have a longevity program, and we're actually getting ready to run a randomized control trial for anti-aging. So we are very interested in that. So we'll see how it goes.
So I need to ask you on that. What are the markers you're looking at? That's like a whole thing, right? What are the markers of aging that you're going to measure? So
we published a paper on aging models, both in the microbiome side and the human side, and we have discovered phenomenal markers of aging, both on the microbial side and human side, and we're currently pursuing those nutritionally. And if big pharma wants to join us to develop drugs, we're open for that. But we're not a drug development company. We're not going to go down the drug development road. But we have fantastic targets. So if they want to partner with us and do that, that's fine. But we are focused on nutritional aspects.
Are you able to tell me what the markers are you're looking at with aging?
Whatever I can tell you is what's published. So that's it. And we have to keep it a little bit higher level than the details, even though it's patented, just so that we don't spill all the beans on the proprietary findings. Okay.
Fair enough.
Yeah.
We'll stay tuned on that one.
Yes.
This is fascinating, Momo.
That's why my life is amazing every day. It's so exciting. So exciting.
Is there anything you want to leave us with?
Well, that depends on what the interests are. We can talk about so many things. I guess, in summary, if people are curious about Viome Life Sciences, like I said, we are a platform company like the App Store. We're looking always for partners, big and small, to work with us and to enable additional applications in human health, but also in other health. So, for example, our technology is absolutely not limited to human health applications. We are limiting our resources to human health applications. But if someone says, you know what, I want to do everything you guys are doing for humans, for dogs and cats, they're welcome to bring in an investment, and we will give them turnkey access to 100% of our technology and convert everything we know into dog and cat health or anything else. And they can go start that company, they can go run it, they can market the products. We will enable them to succeed, and we'll collect royalties from their sales. But we do not have the resources to commit to that. If someone wants to focus on animal welfare or farm animals, or if they want to focus on soil health or water health, ecosystem health, space station health, we don't really care. There's no limit to this technology. We always welcome new applicants, and we have an application process all spelled out on viomelifesciences.com website. So that's one takeaway. Another takeaway is that every person's microbiome can be healthy or it can make you sick. The microbiome you already have is very likely has a potential to be healthy or sick. The difference is what you're going to feed it. If you feed it the wrong foods, it's going to turn into an unhealthy microbiome because it's going to produce unhealthy chemicals. If you feed it the right foods, it's going to produce healthy chemicals that nourish us. And the only way that you can find out which foods are good for you and which ones are bad for you or your microbiome is to do a microbiome test. There is no other, you cannot guess because you just don't know, right? And so that's another takeaway. Another takeaway is that every disease or almost every disease in the human body is a systems biology problem. It's what we call a holobiont problem. Holobiont is the human ecosystem that includes every microorganism on us and in us. And that if we ignore that, we're not going to be able to solve problems. If we don't take into account the entire ecosystem and the environment. And those are some takeaways. Yeah, stop taking antibiotics and stop eating foods with preservatives and switch more to whole foods. And those are some general comments.
Momo, give me your vision 10 years from now, healthcare.
Okay, that's exciting. So healthcare for chronic diseases, cancers, and aging will be distributed to people's homes. Technologies like ours will empower people from birth to death to know exactly the status of their entire body at the molecular level, not at the symptoms level, but at the molecular level. And that's a huge distinction because symptoms are the terminal stages of a disease. Molecular disease happens years or decades before the symptoms are felt. And if you want to know your status, that you're heading for a heart attack, you will know that 30 years before the heart attack. And you will know via the app exactly what to do about it. And your risk, there's going to be a gauge for your risk, not for the average risk of people who are similar to you, but your risk. You will get, this is the way I imagine it. Think about an airliner that has all those different gauges, right? We have developed a system for airliners that are super complex to do preventative maintenance on them so that very rarely does something go wrong. If something does go wrong, an instrument immediately goes off to the red area and shows you an alarm saying, whoa, this system is wrong. And then the checklist that you have immediately tells you what to do about it so that the gauge goes back to green, right? That's the vision for human health, is that we're going to have gauges on our app that are based on our tests. As soon as the gauge goes from green to orange, even before it gets to yellow, the app is going to tell you, hey, pay attention. You're developing a high risk for diabetes. You need to really stop eating those bad foods that are bad for you. I need to start eating more of these foods that are good for you. And it'll coach you through that, right? And it'll include not only nutrition recommendations, but exercise. Do you need to walk? Do you need to run? Do you need to lift? Do you need to do high intensity interval training? Do you need to do gentle walking? What time of day? Is it morning, noon, lunch, dinner? How many times per week? It will include sleep. When is the best for you to go to sleep and how long do you need to sleep? It'll include all of those things. So basically, Viome app in 10 years will be like Alexa or Google Home, where it'll be a coach that's going to talk to you, and it's going to be enabling you to cook healthy food for your family, to make the right choices every time that you're in a setting like a party or a restaurant, to make the right choices about exercise and sleep and supplements. It'll guide you basically through life from birth to death so that you have an extraordinarily healthy life and likely an extended life as well.
Sign me up.
I know. That's what we're working on 7 days a week. That is the vision we're working on 7 days a week. We never stop, never stop. First thought in the morning when I wake up is, what can I do today to accomplish that mission?
Amazing. I love your vision of the future, and I want to be part of that. I'll tell you. This has been vastly informative. I want to thank you for from my gut what you've done. And
just getting started. We have a lot more work to do, but yes, you're welcome. Thank you. I'm glad to hear testimonials like yours. We get those all the time, and it's super, super inspiring.
Yeah. Thank you so much. I know you're a busy guy. Appreciate your time.
Thank you. Thank you, David.
Take care, Momo.
You too.
Bye now. Bye. Wow. Was that ever a knowledge drop? If any of you have questions out there, you want follow-up, you want to learn anything else about this technology, about what Viome does, about RNA, about anything, please hit me up, david@superage.com. Chances are I won't be able to answer that question, but I'll be able to hit up either Momo or somebody who works with Momo and get your question answered. Really, really exciting what's going on out there. I have a feeling, we hear this all the time, like, oh, well, there'll be flying cars. I mean, I've been hearing about flying cars, like, I don't know, since the Jetsons. I don't have a flying car yet. But I think with this area, as far as health sciences go, with wellness, I really believe that the era that we're sort of exiting is just going to be looked at as like medieval in a few years. We're all going to be much better off for that. Everyone, I want you to have a wonderful week. Please enjoy the last few weeks of August, wherever you are. And we're going to see you next week with another great podcast. Take care now.